This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 927, recorded on August 12th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everyone. Hello, Vincent. The weather outside is spectacular. I said yesterday on TWiP that the weather looked nice, but you didn't want to go outside. It was hot and humid. Today, the temperatures have modified down into a reasonable number, and the humidity has gone down also, and it's absolutely stunning. And here I am sitting inside.
Yeah, you're going to be there for the next 2 hours, Dixon. Sorry.
That's
also
okay.
Joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here, it's just about perfect weather. Also, 78 Fahrenheit, 26 Celsius. Just a few white puffy clouds in a gorgeous blue sky. It's been cooling down in the evenings. It's just great.
And there you are indoors as well.
Mm-hmm.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 99 degrees. And, you know, a little bit of overcast. And with, this is important, many fewer 100-degree days in the long-range forecast. So maybe we are exiting this craziness. I don't know, but I could sure do with some rain.
We have a guest for you today from U.S. Department of Health and Human Services, Michael Mercelinski. Welcome to TWiV, Michael.
Thank you. And I'm from just outside Washington, D.C., and sharing the weather described earlier. It's probably one of the 10 best days of the year.
Yep, that's how I'd say it.
So, Michael, you're at BARDA, is that correct?
That's correct.
All right. Biomedical Advanced Research and Development Authority, which is also outside of D.C., is that correct?
Yes, the actual headquarters are down near the Humphrey Building inside of D.C.
So when you go to work, you're at home now, but when you go to work, that's where you would go?
Yes, my office actually looks out. I can see the Capitol from my office.
Oh, nice.
And all the people?
I wasn't there.
If you want to call them people.
Interesting. All right. Well, we have you here to talk about, of all things, monkeypox and what can be done in terms of vaccines and antivirals. But I wanted to give people a little sense of your career. Let's start with, where are you from originally, Michael?
I was born in Washington, D.C.
Wow.
Went to college at the University of Maryland and then moved up to Yale University for grad work and came back to NIH for my postdoc. So I'm a homebody in a sense.
Wow. So you University of Maryland, was College Park?
Yes, sir.
Okay, where ASV was, Kathy, not too many years ago, right? That's a lovely campus.
Yes.
Although we had to do a lot of walking to get around, right?
Well, that's how you got to see the lovely campus.
Yes, that's right. As opposed to Wisconsin, where we're all in one building.
Right.
In college, were you a biology major, Michael?
I majored in general sciences. At the time, it goes back far enough that it was the beginning of molecular biology. And so I took courses from many different disciplines. I actually started as a chemistry, physics, math major and ended up in biology. And so I had to put together a major, which was sort of a combination of science, which then allowed me to get into graduate school.
As I recall, the University of Maryland had one of the finest physics departments in the world at one point because Spiro Agnew recruited all of them and wanted to brag that he had. Were you there during that time?
I was there I took some biophysics courses and I did take some physics courses in that. Their primary, I think, claim to fame was in the gravity area, and I didn't take courses in that area.
So you're saying you're not well grounded, huh?
No, it was pretty heavy.
Not attractive.
So what was the what got you interested in science? Was this something in your family or something else? Do you know?
I was basically a STEM person, and when I got to college, I took some basic biology courses and found them fascinating, and then immediately jumped into courses at that time. We're talking about Mercelinski and how different approaches were used to solve problems in molecular biology. It was really at the very beginning. There's a book called The Eighth Day of Creation. I don't know how many have read that, but it sort of explains the excitement of taking biology, which was an observational science, and turning it into an experimental science. And really, a lot of it was occurring at that time. And so it was very exciting. Every week, something new would be coming out in Cell with regard to developments in molecular biology. So it was a very fruitful time, and I got excited by that. That's why I pursued it. So
when you went to Yale, what did you do your PhD in there?
It was molecular biophysics and biochemistry department. It was they had a combination. It was some crystallographers and some virologists, and I was on the virology side.
You didn't happen to see Rich Condit at Yale, did you?
He got out just before I got there.
But Michael inhabited the same corridor in the basement of the Sterling Hall of Medicine where I was, so we had a very familiar environment. And Michael's PhD advisor, David Ward, was on my thesis committee, so we have a lot of overlap.
At that time, I was a competitor with Vincent in the sense that I was trying to clone minute virus of mice at the same time you were working on polio.
Oh, I didn't know that. I was oblivious to everything, which is probably good. I was going to say I met David Ward after I went to Columbia. I think he came and gave a seminar. Yeah, minute virus of mice, I recall. In fact, Marie Chow, who was a postdoc with David Baltimore about the same time I was, she did her PhD with. Did you know Marie? David Ward.
Yes, we did. We overlapped for a few years.
Who unfortunately passed away last year. I'm going to her memorial ceremony in Little Rock next month to give.
Did I know that? God, I must have known that.
That she passed?
Yeah, but I had
probably
forgotten.
Did. Yeah, you probably did.
Whoa. Okay.
Anyway, so then after Yale, you went to NIH to do a postdoc with who, Michael?
Bernard Moss, who was working on pox viruses and vaccinia, and he was developing them both as a transcription system and also studying DNA replication. So I concentrated on basically the processing of the ends during the replication and those steps which are required to make new genomes. So that was sort of a minute virus of mice, which was among the smallest viruses, to starting to work on among the largest viruses that were known at that time.
But interestingly, when it comes to replication, they have features, at least theoretically, in common.
Right.
Because of the hairpin ends.
The hairpin ends, right.
The model for replication of pox viruses is based on the parvovirus model of replication.
Rich and I went to do a TWiV with Bernie in, what was it, March 2020, Rich? February?
No, it would have been, it was pre-pandemic.
Yeah.
Okay, March 2020, nobody was going anywhere. Okay, so I'm thinking it was 2019.
No,
unless we squeaked in right before things shut down.
No, we did. It was 2020. We went Fred Murphy and then Bernie Moss.
Yeah, that's right. That's right. I remember talking to Fred about how a card game at a retirement home would be a great super spreading event. So COVID was present, but we hadn't quit traveling yet.
Yeah, so in fact, we did a recording in the hotel room, if you remember, talking about what was going on.
Yeah.
Anyway, Bernie was a lot of fun. That was a great episode. So then you finished your postdoc. What did you do after that, Michael?
Well, from there, I worked for a little while at the NIH and then moved over to the FDA and worked in the Center for Biologics, worked on vaccines, and a lot of first of all, the vaccines that were using pox viruses as vectors, and then later, as it became more important, the actual vaccines that were being redeveloped for smallpox. And, eventually they also have Jynneos for monkeypox as well. So that was the area of the FDA I was in. And after 9/11 and after a few more years, when BARDA was created, I was recruited by someone I knew at BARDA who said that if I moved to BARDA, I could be more proactive rather than reactive and start helping people develop products rather than waiting for submissions to come in. And I found that very challenging, and so I moved to BARDA about 2 years ago.
So relevant to your background here, of course, relevant to today's discussion. So you are 100% at least pox virology, and you spent a significant amount of time in the FDA in CBER. And CBER, refresh my memory, it's a sort of you wear two hats, a reviewer and a scientist, correct? That's
correct. We have a lab and do investigator-initiated research, but at the same time, we're responsible for submissions from companies and evaluating vaccines, in this case, whether or not they are appropriate, what tests should be designed in order to make sure that they're both safe and efficacious, and in terms of moving these vaccines into the modern world in terms of manufacturing and evaluation.
So you're intimately familiar with the inner workings of the FDA and the vaccine approval process. You were even part of it, right?
Yes, actually, I was the chair for the licensure of ACAM 2000, which means that I'm responsible in signing for that. And I was also part of the team that was evaluating other vaccines that were under development at the same time.
Okay.
Now, tell us a little bit about BARDA. You said it was set up in response to 9/11, and so what was the idea?
BARDA was created in about, I think, 2006 by congressional legislation that recognized the fact that products, for example, were being developed at the NIH or by sponsors, but they were having a hard time getting them across the regulatory path to licensure, and that there were a lot of threats such as anthrax, botulinum intoxication, smallpox, where help was needed to get these products through the regulatory process so that they could be made available to the American public. And so BARDA was set up, and it was, the term that was used was called the Valley of Death, which means the time between phase 1 and licensure, which is where most products fail. And so BARDA was created, and it has a collection of specialists, some who are manufacturing, that come from, for example, Merck or Pfizer and so forth. They have ex-FDA for regulatory advice and experience. And they also have people who manage the contracts, because the way BARDA works is they award contracts to sponsors who may have a product which is usable, and then we support the development of that product both through money and through our expertise.
So am I correct then that the main cause of death in the Valley of Death is not being able to get the expertise and the money together, in particular the money, to conduct a phase 2 trial?
That's
and beyond.
Right, and that's a lot of the rationale for establishing BARDA. And as well, there's, as you know, from a product, it's not only just the science behind it, but there's manufacturing, and there's meeting regulatory requirements, and how well do you develop assays, validated versus qualified. These things all are part of the evaluation process, because the FDA holds products to a very high standard in order for them to be approved and usable by the American public. So Rich, is both money and expertise. And the way that BARDA was put together, it was a collection of individuals who could deal with those problems.
So this is really important, because otherwise you're really got a big problem getting stuff moving along.
Yeah, and BARDA has been responsible for the licensure of over 60 products now, and some of them are diagnostic, some of them are small molecules and antibody-based products, and some of them are vaccines. But wherever a threat is identified where the marketplace doesn't serve well, BARDA doesn't develop heart medication or things where the marketplace can take care of what the problem is. But there's no real urge to make a smallpox vaccine or a monkeypox antiviral. And so BARDA would support sponsors that could do that.
Michael, does BARDA at all interact with DARPA?
Yes, we have strong interactions with DOD and NIH and FDA and CDC. And a lot of times we have shared meetings with DARPA in order to discuss different products. And sometimes they can be very good in the sense that we have shared goals, and sometimes the goals for the DOD are different than the goals for a civilian response. And so we have, yes.
So while you were at BARDA, you were involved in the development of the smallpox vaccines and antivirals. Can you just give us a brief history of that?
Sure. Sort of overlap. Part of the time in the FDA and part of the time at BARDA. When I was at the FDA, the first, if we go back to the seminal events, probably the first event would be the discovery that the Soviet Union was making smallpox and using this as a potential bioweapon. It was back in the early 1990s. And at that time, there was the impetus was to start making a new smallpox vaccine. Because at that time, the smallpox vaccine was called Dryvax. It's probably the one that you may have received when you were young. And it was basically what was called calf lymph. And what would happen is you'd dip a needle into a virus, and then you'd scrape it across the stomach of a cow, and you'd make a set of parallel lines, and you'd wait for the infection to fester, and then you'd scrape off that material, and that was your vaccine. And so there was only a limited number of doses, and the last manufacturing was back in 1980. So in the early 1990s, there was a push to say, can we develop a new vaccine that's based on modern manufacturing methods and could be tested and demonstrated to be effective, but at the same time not be a vaccine that's basically scraping material off the stomach of a cow. So that was ACAM 2000.
It's a terrible waste of an education to come to that point in your life.
Well, it actually turns out a cow, you can get about 400,000 doses. So,
well, that's called a cash cow, by the way.
Well, and that, that vaccine eradicated smallpox. So it's
that's right.
No small accomplishment.
I think it's Jeff Smith who's always quick to point out to me that the only disease was eradicated by a vaccine that was never approved by the FDA until after the But so that was the impetus for beginning of ACAM 2000, which is a tissue culture clonal isolate of that Dryvax vaccine. And as after 9/11, there was a second sort of burst of impetus, I would call it that, is that we went from a small stockpile to a large stockpile. And so that's what we have now in terms of the ACAM 2000. And that's basically a vaccine which is the same as your father's vaccine, so to speak. And it's a very effective vaccine. And as Rich pointed out, it was very effective as a ring vaccination approach to eradicate smallpox. The drawback to it is that it's a live replicating vaccine. And because of that, it's associated with adverse events, especially in an immunocompromised population, and can result in very serious adverse events, including death. And so the risk-benefit for its use as a prophylactic or in a low-risk environment is not low. But if there ever was a real outbreak, it's reassuring to know that we have a large amount of a vaccine which we are extremely confident would be very effective.
So the risk here is an important part of that quotient.
Yes.
And a smallpox outbreak has a different risk assessment probably than the current monkeypox outbreak.
Right,
correct?
and that's exactly right. And so one of the early attempts to say develop another or a next generation smallpox vaccine was where Jynneos comes from. And that was an attempt to develop something that was safer and more universally applicable than ACAM 2000. And not necessarily to replace ACAM 2000, but to be an adjunct during a response so that you could now get vaccine into more people. If people that you weren't sure of or you were worried about because they were associated with adverse events would get Jynneos, whereas most of the population would still get ACAM 2000. And Jynneos is MVA-based for those who know the field, and MVA stands for Modified Vaccinia Ankara. And to back up a little, during the eradication campaign, there were many different strains of vaccine that were used. Pretty much, depending on where you were, you used a local strain. In the U.S., it was mainly New York City Board of Health. In Europe, it was Lister. But one of the ones that was also used in Europe was Ankara. But Ankara was associated, as were the others, with a sufficient number of adverse events that as the number of cases dropped, it became clear that the risk of vaccination now was just as bad, if not worse, than the risk of disease in most of the world. So there was an attempt scientifically to develop attenuated vaccines, and they were done by passaging the virus through tissue culture. In the case of the MVA, it was through choriotylic membrane and egg cells, CEFs cells. And what happened was they would pass through CEFs, they would harvest that, pass it again. And after over 500 passages, they had a virus now, which was MVA, which no longer was as potent as the Ankara starting material, grew extremely well in chicken embryo fibroblasts, but did not grow well in human cells or other cell lines. So now you have a virus which you could prepare in chicken embryo fibroblasts, which when you give it to people will be self-limiting and safer than the other vaccine. And that's the sort of rationale for Jynneos. What Jynneos is that vaccine.
And it is administered differently because of its properties, correct?
Right. I mean, the vaccine, the ACAM 2000 is a scarified vaccine where what you do is you take basically a two-pronged needle and you break open the skin and deposit a small dose there, which then replicates and forms a lesion, sort of like a pustule, which we're seeing now, for example, with the monkeypox. Jynneos is actually a subcutaneous or intramuscular injection, like most vaccines are given. And it's because of when they went through its evaluation to the FDA, it was clear that two doses gave you a prime boost regimen, which gave you a much more robust immune response than a single dose. And so that was the accepted regulatory path for development of Jynneos, two doses, 28 days apart.
So you mentioned these are both part of the strategic national stockpile. Is that what is that? Is that just vaccines or other things as well?
Yeah, no, the Strategic National Stockpile is basically a storehouse. It's in a couple different locations in the U.S. It has vaccines, it has antivirals, it has PPE, it has antibiotics, it has the number of different items that would be needed in a response. For example, if there was an anthrax attack, you'd need a completely different set of countermeasures than you would for a smallpox. So in those those storehouses, there's also provision to store at -20, and there's things stored at room temperature and so forth.
So roughly how many doses of ACAM 2000 and Jynneos do we have there? Is that known?
I don't know if I'm supposed to say it. We have a lot of ACAM. Okay. I'll say that. With Jynneos, it was a slightly more complicated story in the fact that it has a expiry period of about 3 years in its liquid frozen form. And so we realized that we couldn't replace the stockpile every 3 years. It would be just too expensive. And we've pushed the development of a lyophilized form, which is a freeze-dried form, which preliminary data indicates it has a very long half-life. It's going to have a much longer expiry period and will be the appropriate way to go. But we don't have that formulation licensed yet. So what we've done in BARDA is we've stored the intermediate, which could be converted to the lyophilized form when it's licensed. But we're now using that to fill doses that can be now mobilized during the monkeypox crisis, because the liquid frozen form is licensed. That was shown to be effective.
So check me on this. My understanding, without any hard numbers, is that there's a lot of ACAM 2000 stored away and significantly less Jynneos. But the go-to vaccine in this particular circumstance, given the risk profile, is Jynneos rather than ACAM 2000 because of the potential side effects of the two. Is that a fair statement?
I think that's absolutely correct. I think you always have to measure the risk-benefit, and that one of the reasons for Jynneos as well as ACAM is to give us flexibility in response. And in this case, because it's a high transmission, low mortality virus, then what you can and should do is different than a high mortality, high transmission virus. And so, yes, Rich, I think you're absolutely correct. We have to measure the consequences of using each vaccine as well as the benefit.
So I'm thinking that in the short term, this issue of licensure of the lyophilized Jynneos is not really an issue, right? Because
no,
you start with the same stuff and you just freeze dry it, and what we need is what we got or are making now. And the freeze-dried thing would be more for a long-term storage solution. Is that correct?
Yes. Yeah. Now, okay. All of the necessary clinical trials have been done to demonstrate that they're indistinguishable. So that part's finished. But the part that's been delaying the actual licensure is the manufacturing side.
I see.
Because we have to put in and demonstrate that we have control over the process of lyophilization and storage at different temperatures. So,
you know, it's interesting because every time I think about this bit of licensure, I don't think about the manufacturing stuff, but that's got to be, in terms of licensure, every bit as important as anything else, right?
Yeah, and I actually think, you know, back to tooting my own horn, is that the BARDA, I think when it was created, one of the smartest things they did was to incorporate a lot of the manufacturing experts into the group, so that when we come up against something like this, a lyophilization process or what manufacturing problems exist, these people have actually dealt with this before. So in that case, you know, we're able to solve problems that NIH, CDC wouldn't be exposed to because they don't have that experience.
So in the absence of having any smallpox, how were these two vaccines tested? You mentioned immune responses. Is that what was measured?
Primarily, what the FDA did was what's called, we did bridging, which is a fancy way of saying comparing to what we had confidence in. So DryVax was actually used in the eradication campaign. So that was used as the gold standard. And when ACAM 2000 was manufactured, it was compared to DryVax, both in terms of take rate, which is the lesion that's formed when you're scarified, as well as immune response into those that were vaccinated and those that were not vaccinated previously, and shown to be non-inferior in measurement of ELISA, which is binding antibodies, and neutralization antibodies through a PRNT. And so that was considered to be sufficient evidence for efficacy. And then the safety was, of course, done through clinical trials and shown that it was indistinguishable from what was DryVax. Jynneos was a little more of a different problem because it doesn't form a lesion. As Rich indicated, it's actually a subcutaneous or intramuscular injection. And so it was based on solely the immune response, and in two ways. First of all, the ELISA and PRNT values, and after 2 shots, the 0 and 28-day prime boost regimen, the immune response as measured by ELISA or PRNT is higher than that which is established by ACAM 2000 in the same time. And so that was considered to be sufficiently proof of immunological efficacy, if you want to call it that. And there was also, because the trial was run in the military was required to be vaccinated with ACAM 2000. And so a small cohort was vaccinated with ACAM 2000 after they had received Jynneos. And the rationale behind this goes back to, if you've been vaccinated and then you're revaccinated, you get a modified or much smaller take because you already have an immune response which is considered to be sufficient to be protective. And so in this case, when they came back and revaccinated people with ACAM 2000 who had already received Jynneos, a great majority of them had either a modified or no take at all, and a small percentage had an indistinguishable take from those that had not been vaccinated.
So to be clear here, a central issue is that the gold standard for the effectiveness of a vaccine would be, and we're going to get onto this with the drugs, I think, as well, would be that it protects humans against the illness that's being targeted, which you could do with TWiV because smallpox was present and you could show that it prevented smallpox. But with smallpox gone, you can't do that anymore. So what you have to do is use other indicators of immunity and compare these new things to TWiV.
Right. And so we call it bridging, but yes, exactly that.
Yes,
but
that's bridging. Okay, good.
Right. But also point out there was other supportive data that was done. And for example, testing in animal models that were challenged with monkeypox. And at the CDC, they did actually live variovirus newts and ELISAs and showed that the vaccines, that the immune response engendered by the vaccines was sufficient to protect against that. None of it is direct protection against the disease, but it's a sort of, collection of data, some which is, for example, the immune responses in terms of ELISA and PRNT would be held to a very high standard in terms of the assays and in terms of validation and demonstration. Now, supportive data may not be held as high in terms of assay reproducibility, but still the data would be important to demonstrate that the activity was consistent with what we'd expect for an effective vaccine.
So I have a way of summarizing this, particularly as I read the antiviral paper, and that is that smallpox was eradicated in part because no animals are infected. And
that's
those
the animal
two
reservoir.
facts,
Yes.
and those two facts are the biggest challenge for developing antivirals and vaccines. That smallpox has been eradicated, so it's not out there to get, to test something against natural infection. And also no animals are, naturally infected. And so that's the second part of the challenge to me, anyway.
You actually bring up a very important point that wasn't as important for the vaccines as it was for the antivirals, which is establishment of an animal model where you can test your product in the absence of actually challenging human beings. And for many of our products, for filoviruses like Ebola and Marburg, and for anthrax, and there's animal models which have the same sort of pathogenesis and response to the disease as humans. And so you can test your product in those models and try to develop what is the appropriate dose and what is the appropriate mechanism and what is the, how will it look like if this is effective or not under the circumstances you want to use it. But in the absence of an animal model, the bridging becomes much harder. And your point is actually very important because it's impossible to directly do what we'd like to do in terms of what we can do with other products.
So do we know if these vaccines, ACAM 2000, Jynneos, would protect against monkeypox infection?
The data on ACAM 2000 and its progenitors, Dryvax and so forth, is there's an estimate of 85% or higher because of its use during the campaign to eradicate smallpox. I think also if you look at the cases in Africa and its rise of endemic monkeypox, most people associate that with the waning of immunity because people aren't vaccinated against smallpox anymore. And I suspect that it's very highly effective against monkeypox. Now, we have tested the animal models. We can't use variola, but we have tested them with high doses of monkeypox that would be a lethal challenge model, and they protect. They're excellent as far as providing protection in those animal models.
So for monkeypox, it's a non-human primate model, is that right?
Yes. And
do you think that, given the current numbers of monkeypox virus cases, that there will be a clinical trial done for efficacy, or is that not needed?
For Jynneos, the label has an indication for monkeypox, so no clinical trial is necessary for its use against that population. There is, if you've probably noticed, because of the limiting amounts of vaccine, there's discussions about applying it in different ways in order to get a better immune response. There was some preliminary data done that showed that an intradermal, which is injecting it just under the skin, similar to scarification, would give you a better immune response than the the intramuscular or subcutaneous. And so there's a trial that the NIH is setting up to look at that, which would spread out or stretch the amount of vaccine because you could then use lower doses to get the same response. The antivirals, when they were licensed only against smallpox. And so CDER, which is a Center for Drugs, is looking to have a clinical trial as part of its deployment of the TPOX from the Strategic National Stockpile.
The TPOX now, however, can be and is being deployed for use in the monkeypox outbreak, correct?
Yes, but again, it does not have a label indication for monkeypox. And so for its use against monkeypox is not licensed. And so the CDER is looking to have some clinical trials in order to get real clinical evidence of its efficacy.
So does the lack of licensure then create a barrier to its use in this circumstance? A practical barrier?
It creates a practical legal barrier in the sense of what's protected under law and what would be, in terms of the PrEP Act, pays people for adverse events associated with the use of the products from the Strategic National Stockpile. But the recent declaration of an emergency drops most of those barriers.
Okay, good.
So I have a question because this perspective that Vincent gave us from New England Journal of Medicine talks about that there was a randomized controlled trial that was about ready to be launched in the Democratic Republic of Congo to test the Tpox, and that now NIH is developing a U.S.-based one for this newer worldwide outbreak. And so I wonder how much of a concern it is that these are from two different clades. How different are they, and do we know much about the difference in response to Tpox?
What I'll say is in vitro, in tissue culture, it's equally effective. And part of it has to do with the way that pox viruses are organized. The central portion of a pox virus is where all the housekeeping genes, where the DNA polymerase, the RNA polymerase, the proteins that are basically involved in metabolism of the virus. And those are widely conserved amongst pox viruses, which is why the vaccines seem to protect against many different orthopox viruses, and the antiviral will be protected against many different ones because the sequence doesn't vary much in that region of the genome, which is where the target for Tpox is.
So maybe you could briefly review for us the development of this antiviral. So we have the development of the antiviral Tpox, because that involved this development of satisfying a two animal rule, okay, or using animal models and trying to figure out which ones were appropriate for approval of the drug. But I think importantly, a lot of that also included some testing with monkeypox, right?
Yes.
So maybe you could go over some of that.
Could you just say what a T stands for before you go any further?
in the Tpox?
Why are they calling it Tpox rather than Fpox or Gpox? Or what's the T stand for?
I don't know. It's original name. I'll take this.
I'm sorry. I thought I was the only one on this podcast that didn't know what a T was, so don't worry about it.
Well, its original name, it started out as ST246, which is just the name from the screen, and then it was called Tecovirimat. So I'm going to tell you the T is from the Tecovirimat.
Okay, Tito is fine.
Even though I don't know, I will claim that.
Where did it actually come from?
It came from a screen. What happened was, especially after 9/11, NIH started supporting screens of licensed compounds and unlicensed compounds in vitro against pox viruses to look for potential antivirals. And this, moiety, this sort of backbone came up in one of the screens for. Basically, it was just a chemical screen. It wasn't a licensed product. So they went through a period of optimization where they did chemical modifications to find the best in terms of the structure. And then it was then moved into development in terms of testing in different animal models. And I think, this is a point that Richard brought up and was brought up earlier, was that how do we test it against variola when we don't have an animal model with variola? Because the problem with drugs in terms of development is it's all about exposure. What's the concentration in a sick individual? What's the concentration in a healthy individual? And can you get a sufficient concentration in order to have the effect that you want? Vaccines in some ways are much easier because you can measure the immune response. But how do you measure the exposure in an infected animal when you can't infect that animal with the virus that you want to test it against? And so it poses a problem which the FDA addressed by creating what they call the animal rule. And the animal rule allows efficacy evaluation to occur in animals rather than in clinical trials. Now, you still have to go through the same safety in terms of the demonstrating its safe number of people. You still have the same level of manufacturing scrutiny that you would for any other product, but the efficacy can be based, again, on animal data. And this, again, requires a certain amount of bridging. And there's not a hard and fast rule that says if you get to this point, it's now licensable. Basically, you accumulate information till you get to the point where the FDA feels they can make a regulatory decision. And it's a difficult decision for them to make. Now, for TPOX, as Rich indicated, there was a lot of work done with monkeypox challenges in that non-human primate model that I talked about earlier, demonstrating through a dose-response curve that low doses didn't work and higher doses did, and finding out what exposure was required to get protection in that model. There were delays so that you delayed the administration of the drug farther and farther until essentially the drug failed. And then there was also the shortening the time course, because it's usually given as a 14-day, every day you take the pills drug. And that's just because of its turnover time in the body. And so with all that data, you then go to the FDA and try to put together a rationale for what dose you like and why that dose is appropriate. And then you demonstrate in animals that you can have a certain exposure, and then you demonstrate that in uninfected people you can reach that exposure and what corresponds to that dose. And then, of course, the only thing you don't have is infected people. And that's part of why I think the FDA is so highly scrutinizing what's going on now, because they want some of that PK data from people who are actually infected with monkeypox in order to make sure that all the bridging they've done to get here was appropriate, because there's a certain small level of uncertainty that's still involved.
Absolutely.
Because you don't have direct demonstration of efficacy. So I appreciate how difficult it is for the FDA to make these decisions. And, from a BARDA perspective, of course, we're pushing to get those decisions made, but it's not an easy decision to make.
So Koch's postulates.
Yeah, exactly.
We're stuck with Koch's postulates because of Koch, but there's a ton of organisms out there that if he had worked on them, he would have never developed them because they're very specific for each host. And obviously some of these viruses are also.
Right.
One little footnote I want to make here, because people are reading the news, and the namings can be confusing. So ST246, tecovirimat, and TPOX are all the same thing.
Yes.
Okay. There's one drug. Actually, no, this raises an issue. There is more than one drug available, right?
The second drug was only recently approved, and we do not have any of it in a strategic national stockpile. We're presently negotiating a contract with the company, but it was so recent that we never had the chance in order to acquire any.
And that's the orally available cidofovir, is that correct?
Yes, it's brincidofovir, which is it's basically a prodrug of cidofovir. It's a fatty acid attached to that, which changes its biodistribution, so it doesn't have the same renal toxicity that cidofovir has. And then it's basically a nucleotide analog after it's cleaved, and that nucleotide analog is phosphorylated, and of course ends up in the metabolism.
And there's a lot of TPOX in the stockpile. Is that correct?
Yes. There's more than enough TPOX in the stockpile.
Okay. Is TPOX being used now or not?
Since the declaration of the emergency, it's being mobilized and used. Yes.
Okay.
And as I said, that they also are trying to collect clinical information because that clinical information doesn't exist, and to use that as and I think it was earlier mentioned of how there was going to be a trial in the Democratic Republic of Congo, which is correct. The WHO meetings, there's a WHO meeting every year on live research with live variovirus, because any research using live variovirus has to be gone through the FDA, go through the WHO. They approve with a committee that they have, which is the international group of scientists. In the last few years, the products that are being developed that are against smallpox, which is, of course, Jynneos, Tpox, and so forth, it was very clear to that group that they're also, or should be, effective against monkeypox. So there's been a push to try to study them in Africa to get more information. And that's what that trial was being set up in the Democratic Republic of Congo through the company SIGA, which makes Tpox, with, I think it was NIH and London, school in London.
So I might have missed it. How is Tpox administered?
It's a pill. It's a capsule, technically. It's a capsule, and of course, it's orange and black because Dennis Ruby, who developed it, is from Oregon State.
Nice. That part of the mythology I'm familiar with.
So I think it serves as both advertisement and antiviral at the same time.
God,
right. So how many pills do you have to take before you're completely protected?
It's a 14-day regimen.
Oh, I'm sorry. You did say that. You did say that. That's
and
right.
It's twice daily, 14 days.
I got confused because I thought you were referring to a virus. Tpox. Well, pox. What kind of pox is that? It's a pox on my house.
The FDA names everything when it gets licensed, and that's the name they chose.
Very confusing for our listeners.
So I wanted to get. Go ahead. I'm sorry.
I was saying we've called it many different things over the years because for a while it was ST246. For a while it was tecovirimat. I mean, Jynneos used to be Infimune, and so.
Right.
Well,
so many people are upset that there's not enough Jynneos, right? And we touched on earlier, the 3-year life, and now you have to get through the lyophilization licensure and so forth. So when do you expect that the supplies will be more available?
We are hoping to have more than enough by the end of this year.
Okay.
The filling, which is to take the material which we already have, is relatively straightforward. There's no manufacturing hurdles usually in that step. So there'll be doses over the next few months that should be, I can't tell you how many, but it'll be hundreds of thousands, if not 2 million. And so that should be, well, hopefully more than enough to take care of this outbreak. And then we will, of course, now start to make more material in order to start establishing a stockpile that we can have in case there's another outbreak in the future.
So if you've had monkeypox once, can you get it again?
I don't know. I know that for smallpox, the general dogma was that if you ever caught it, you never caught it again. Right. If you were vaccinated, you were protected for many years, but not forever. So my assumption is that if you catch monkeypox, it's probably the best vaccination possible when you survive.
Right.
So, what was I just going to ask you? So FDA just gave an EUA for intradermal, right?
That's the one I was talking about where you inject it just under the skin. It's there's one of the things we used to talk about with regard to ACAM 2000 or the scarification where you poke with the needle is that you're actually vaccinating in the skin. The skin, of course, is a very important organ and it has a lot of immune response because it's our first layer of protection. It's much easier to administer by subcutaneous or intramuscular, just from a practical standpoint.
Right. So
that's the development path that was used for Jynneos. However, one experiment that was done during the development path compared this intradermal, which is it's used for tuberculosis testing, where you just go right under the skin. It's an acquired but but it is a possible skill that showed a higher immune response than normal subcutaneous or intramuscular. So what's being proposed is taking vaccine doses now and splitting them into 5 and doing it intradermally, measuring the immune response and, of course, the efficacy as we can through this outbreak, and seeing if that's a way to sort of stretch out what we have today.
Do you think the difference might be due to the fact that the skin has a lot of dendritic cells, whereas the subcutaneous or intramuscular,
that's exactly what
the pathway is not there?
Now, that's exactly the hypothesis, is that the dendritic cells are really your target. There's an experiment a long time ago where the military was trying to take ACAM, what was it, Dryvax at that time, and administer it in a different way because they didn't like the lesion, because the lesion interfered with their ability to do training and it was ugly and so forth. And so they did a trial where they did intramuscular injection of what would be equivalent to Dryvax, and it didn't work. And in some cases, when they pulled the needle out, the needle track would leave a lesion, and those did have an immune response. So yes, I think there's empirical evidence. I guess I would call it less than proof, but that the skin is a better response organ than intramuscular.
We've talked about this occasionally in the past and hypothesized. I'm waiting for sort of microneedle patches to become a standard, if you like.
I will tell you that BARDA is investing heavily in alternative forms of administration, including microneedle patches.
Excellent.
We have to. They're still at this early stage where we're testing them because of that very reason. You can imagine a scenario where you want to distribute a vaccine. It would be nice just to hand out a patch.
So the Jynneos is manufactured by Bavarian Nordic, right? Is that the sole manufacturer?
Yes, that's correct.
And so they must be ramping up production. Can you give any insight into sort of a timeline for that? And obviously there's a global demand as well, right?
There's a global demand, and I'm sure that there'll be a enhanced production. There's a limit in a sense because, one, it's in chicken embryo fibroblasts, and it's labor intensive compared to some preparation methods. And the other thing is that it's a relatively high dose, and part of that dose is because it's so safe. And the dose is 10 to the 8th. And so you have to generate a lot of vaccine or a lot of virus to get a lot of vaccine. But I'm certain that they're working on that. I don't know what the numbers are.
Okay. So this is a re-emerging infection rather than an emerging infection because it's already been around for a while. And then it sort of waned and then back up again, right?
Right, and the truth is, it's not monkeypox. It's a rodent pox. We don't know which rodent it is, and what happens is that it gets into the zoonotically, it gets into people, and then we're spreading it ourselves. There was the outbreak you're probably familiar with back in 2003, which was the same West African clade, where it got into the prairie dog population. It turns out that prairie dogs are susceptible, and then Gambian rats were shipped in the same container, and they ended up infecting prairie dogs, which then people handled, and then they caught it.
Michael, are you
worried about? Sorry,
I was going to ask the same question.
I just have a follow-up question, and that was, what's our next worry for a pox virus outbreak? I mean, where is that going to come from?
I'm sure.
Bats? Do bats get pox viruses? Bats? Who knows?
Yeah, no, I'm sure Rich will
Burmese
back me up.
pythons down in Florida. I think that's a great source for these things.
Yeah, I assume Rich will back me up. There's lots of rodents out there. There's lots of.
Yeah, to me, one of the major issues with this outbreak is the possibility of spillback into some sort of non-human reservoir in the Western and Northern Hemisphere.
But I would say the thing that gives me some solace is still this is being spread by direct contact and not respiratory. Right. So as long as that is the primary means of spreading, then it's going to be a low R value, and it's not going to spread very easily. The problem is if it becomes a systemic infection, and then we have a reservoir that we can generate. But, again, it's something I think you're right, we need to worry about it. But at present, the characteristics of the disease are such that we should be able to get it under control before that can happen.
So, Mike, did this take you by surprise? The spread of this, because we were, frankly, honestly, on the earlier episodes of TWiV when this came up, I certainly was saying, it's going to blow over because it's recognizable, not all that transmissible, blah, 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 and you ought to be able to deal with it. And it has blown up much more significantly than I anticipated. What's your experience?
No, I agree. I thought it would be something that would peter out pretty quickly, but it's established at a high enough level now that it appears to be still transmitting. I think what we need to do is follow what happens over the next 3 or 4 weeks. If people's behavior changes, then it will not spread, or the spread will drop quickly. I think the second thing is as we of course vaccinate people who are at risk and have to treat people hopefully with the antiviral, that we'll also be able to kind of snuff out the spread of the disease. But it has gotten farther than I expected it to, in terms of in terms of the number of people. But I think it's primarily a demonstration of human behavior and vector transmission more than it is about the actual characteristics of the disease. Because as you indicate, the R value is relatively low, but at the same time, the behavior is what drove this. If you remember the Ebola outbreak in 2014, the one that actually ended up infecting 20,000 people, there was a funeral, I think, early in the outbreak, which ended up being, I would call it a super spreader, in the sense that a lot of people were there and touched the person who died, and so went back and started all these different infections, focal points. And sometimes I think that's what happened here. And the problem is we didn't get to it until after that event had occurred in this case.
Yeah, and the Ebola thing is an interesting comparison too, because if I'm not mistaken, education about the mechanisms of spread in that circumstance was a significant component of getting it under control, right?
Yes. I mean, we got, BARDA got licensed vaccines and antivirals against it, but they were not as effective as the social behavior, which changed people.
So do we know how this, I'm sorry, go ahead, Kathy.
Well, I was just going to say, I want to go against my normal Pollyanna self and say that the hope that in 3 or 4 weeks we will know if behavior is changed enough, but my guess is that behavior won't change. This is where the virologists have had all kinds of difficulty in predicting human behavior. And I have a phrase whenever I see modeling studies, and I say, human beings do not behave like ideal gases.
That's right.
Title.
Yeah. That's good. That's
good.
good. That's perfect.
But the thing is, Michael, this is clearly now outside of a certain population where it's transmitted by close contact. We have many people who have no sexual activity getting infected now, so it's not clear what they have to change, right?
Well, close contact, I guess, is the first thing. It reminds me of, in a sort of oblique way, I guess, is that if you go back to the early 2000s, the military was vaccinating its troops with dry vaccine and ACAM. And they were vaccinating troops because they were being deployed to areas where they thought the risk was high enough that this was warranted. There were cases of transmission between soldiers who wrestled. There were cases of transmission. And so in those cases, of course, it was very few, and so it didn't continue to spread. And here, it's the same method of transmission. The problem is, how can we limit those events so that now we can sort of put out the fire?
So Michael, let me give you a theoretical. So let's say someone has lesions and they're touching them, and then they shake your hand, and then you touch your nose. Is that a way to get infected?
I think that's, there's the lesions themselves contain a lot of active virus, so yes. There's plenty of evidence from the smallpox days of what's called fomites, and the stories about spreading with blankets and so forth. So it has a certain amount of staying power. So from what you describe, I would say yes.
Okay. Yeah.
Are there any vector-borne pox viruses?
Yeah. Myxoma.
Smallpox, yeah.
Do I know one? I mean,
He said myxoma virus, Dixon.
myxoma. Oh,
Yeah, that's the one. That's the one that they used in Australia to crash the rabbits. That's a different kind of. It's a mosquito, but it's a different kind of. It's a mechanical transmission. Doesn't replicate in the mosquito.
That's called a paratenic host.
Oh my goodness, a new word. I'm going to have to remember that.
Let me write that one down.
Mechanical is paratenic.
So I think you said this, but I want to just clarify. You think the vaccination will ultimately control this outbreak. Is that correct, Michael?
I'll admit one area of unknown here, which has been brought up in meetings we've had, and that's that the vaccines were all tested against normal transmission. And this is because of direct contact might be abnormal in the sense that you get a high dose in a local area. So there is some concern that the vaccines may not be as effective as they would be against what we are normally exposed to, which would be a lower dose.
Okay.
But I think that it's certainly true that you're going to get an immune response and you're going to ablate the disease and you're going to make it easier in terms of recovering. And because the disease as it is now, in many cases, is relatively small number of lesions, I think it'll be effective in most cases.
And you need two doses of vaccine for full protection, correct?
The protection that's afforded with 2 doses in terms of the immune response is much better than 1. But this is a live attenuated virus, and so 1 dose does give you a good immune response, and it does provide protection in many of the animal models we've tested. 2 doses is superior to 1 dose.
Would it be useful to have an antiviral for this virus as a salve that you could actually put on the lesion itself to keep it from spreading to anybody else?
Hey, I don't know. SIGA, the company that developed TPOX, did try to make a lesion, some sort of lotion and a salve many years ago, but they found that they basically were underdosing, and they didn't go back and do it.
Yeah, and my impression is that even though the I've been wondering the extent to which in the monkeypox patients now there is systemic infection of viremia, because certainly there are lots of examples of local infection at point of contact and lesions that spread out from that. But it's not clear to me to what extent a given infection is generalized. Do you have any perspective on that?
That's something I've been watching and trying to figure out. And right now, most of the analysis by PCR, and they can find evidence for genomes in many places. I don't know what that means in terms of actually infectable virus. But I think that I don't see any evidence yet that this is turning into a systemic infection. It still seems to be mainly surface to surface. But there's been some evidence in semen, and there's been some evidence that in blood, although I think, Rich, you understand in blood, I think complement usually takes care of it, and I don't think it's infectious. But if you're doing it by PCR, you're finding some signal. What it means clinically, I don't know.
The PCR samples for PCR are normally taken from lesions. Is that correct?
Normally, yes, and that's where you'll find virus. But there's also some attempt to clinically take other samples now as well.
Does this virus have any escape mechanisms from phagocytosis by, let's say, dendritics or by histiocytes, which allows it to remain in a spot and create a lesion?
There's no evidence that I know of that pox viruses are anything but an acute infection. And the fact that a persistent or chronic has only been observed in immunocompromised animals or people. But with regard to most healthy people, they're really, really a good target for the immune system. The immune system makes over 100 different antibodies to these virus infections, and so it's a race between the virus and the immune system, and the difference between the different pox virus has to do with the interaction of these host range genes at the termini and their interaction with the host immune system, and it's kind of why I call monkeypox a rodent virus, because it requires a pretty high dose to be effective in monkeys, and smallpox is just innately designed to interfere with our immune system, and so that's why it's so effective in people, and why you lose the race half the time. But against the strains that are used as vaccines and so forth, you win almost all the time, unless you have a problem with your immune system.
So I have a question about the lesion. So my understanding is in smallpox, you inhale, reproduces in the lung, it becomes viremic, and then you can have distributed lesions everywhere. So for monkeypox, it's not the same. You would have lesions basically at the point of contact with the virus initially, correct?
Right. That's what's happening now. You're absolutely correct.
Okay.
And so in the case of smallpox, when you're exposed, there's a refractory period where you have no symptoms, but the virus is actually establishing itself and going through initial rounds of replication. And then when it goes systemic and spreads through the body, it gets to the capillaries and then sets up new infections, which become those pustules and rash. In this case, it appears that the rash is because it's an actual infection of the virus where contact has occurred and the virus has been deposited there. That's what seems to be what's happening now.
So is monkeypox virus not present in, say, respiratory droplets, so that wouldn't be a mode of transmission?
Well, I don't know if in Africa, where it's endemic, that there is some evidence for systemic infection, and it seems to be more prevalent in children than in adults, which might be just a reflection of the developed immune system. In this outbreak, I haven't seen any evidence yet for respiratory, but if you had, and this is what theorized for smallpox, if you have lesions in your throat, if you cough, then that would possibly expel virus. I know that there's been some, cases where it has been seen in other areas, both throat and rectum, and maybe it would be, but I haven't seen any evidence for that yet.
So you can have mucosal lesions, in other words.
Right.
Okay.
So, Michael, I've been trying to think of a way to ask this, and when you see news reports of people complaining about the availability of either vaccine or drug, what goes through your mind?
It's been a bit, between this and in a sense the COVID response, there is a, I don't want to call it frustration, but maybe that is the best word. We're doing what we can, but at the same time, I understand impatience because people want, as I say with my children, that instant gratification isn't quite fast enough. And I think that's what occurs here as well. And, I think that I know that we're doing the best we can, and that I'm actually extremely grateful for some of the people here at BARDA who had the foresight to put some of the intermediate to Jynneos at -80 to hold it till the lyophilized form was available, because if they hadn't done that, and I had discussions with this a couple of years ago, is when they made those decisions, we would have nothing today. So, you can't prepare for everything. You do the best you can, and in the end, we have something now we wouldn't have had otherwise.
Well, Michael, thank you for your involvement in the development of these vaccines and antivirals. I think everyone needs to be grateful to BARDA and FDA for having these at the moment, because you can complain that there's not enough, but the alternative is not to have any, or one alternative.
That's right.
And Michael,
go
ahead. Sorry.
Thank you for that. I think that we've all have a scientific background, and it's sort of interesting to be in that interface between what you've learned in terms of your science, in terms of what you need to do, and in terms of what can actually make a difference in public health. And I've appreciated that from being at BARDA. Yeah.
And so being at BARDA is still exciting, isn't it, Michael?
I'm afraid so.
All right. Well, keep it up. And thank you so much for joining us. Really appreciate it. It really learned a lot, Michael.
Yeah, Michael, this is terrific.
Thanks for having me on.
Yes.
You're welcome.
Very good.
If we ever had Paul Offit and him on at the same time, none of us would say anything. I would just listen to those two people talk, because There's a wealth of information sitting out there. You ask one question, you get the entire story, and it's just very nice.
His experience is just awesome. And in this particular case, it's not just the FDA and the BARDA experience, but he starts off from the beginning and through as a pox virologist. So in terms of this particular outbreak, it's just amazing.
It's nice to see he hasn't lost his sense of humor either.
Oh, no, Mike's a great guy.
I'm sure that BARDA had roles in COVID, right? Of some kind.
Oh, yeah. Well, I'm sure. I assume so.
But
yeah,
he wouldn't have been involved, I guess. Right?
I don't know. We should have asked him.
Yeah, well.
Can you get him back?
Next time.
I don't know. But, one of the important points is, my understanding is that, on the heels of the 9/11 event, the mandate went out that we want for all of the, potential bioterrorism agents, we want a vaccine and 2 drugs. Okay. And that, from what, 2000, it's 20 years ago. Okay. Yeah. Has been pursued vigorously. And in this particular case, it's been realized. We have a vaccine and 2 drugs against this. Now, maybe it's not as readily available right now as you want, but as Mike points out, it's far from nothing.
Yeah, yeah. So Rich, when you vaccinate, and what are the neutralizing antibodies directed against? What viral proteins do we know?
Oh man, you got to get Shane Crotty on again. Okay?
Oh, he knows this stuff?
Oh yeah. Oh yeah. Shane and Alessandro Seti have done a lot of work on pox viruses and the immunity to pox viruses. Okay. I think, however, your question brings up something that I've been wanting to say, in particular for the newer people in the audience. And this is one of the things that makes virology so fascinating. There's so much variety in virology. Mm. Not all viruses are the same, okay? And you could hardly have 2 viruses that are different than monkeypox and SARS-CoV-2.
Yep.
SARS-CoV-2 is a small, single-stranded RNA virus, okay? Monkeypox is a large, double-stranded DNA virus. They transmit differently. Their genetics are totally different, okay? So whereas RNA viruses mutate a lot and you get all these variants that come up and you're constantly battling the variants, pox viruses have a very much lower mutation rate, DNA viruses in general. And so genetically, they're quite stable. I don't anticipate that we're going to have a lot of variation showing up. And of course, the transmission is quite different. And this goes back to your immunity question. In the case of SARS-CoV-2, in terms of the antibody response, at least, it seems as if just targeting the spike protein does the trick. Mm-hmm. Pox viruses have like 20 different proteins in their envelope. Okay. And I'm not sure that I don't recall right now which are the immunodominant antigens. Okay. And it's also not clear to me that in terms of the specific immune response, we really know what the appropriate targets are. And people have tinkered with trying to make subunit vaccines, mm-hmm. but with no success that I know of so far, because you can't really identify what the appropriate antigen is. It's probably a collection of antigens. Okay. And probably the T cell response has a lot to do with it. So the what do we call it? Correlates of protection, okay, are going to be extraordinarily different, even in general, relative to SARS-CoV-2. It's a completely different picture.
Got it. Okay.
And yes, Alessandro and Shane, they'd be able to tell you.
Because many people have asked, well, why doesn't Moderna or Pfizer just make mRNA vaccines?
I don't think that's happening with this dude. Okay.
Yeah.
You're talking about something that comprises the whole virus. Now, maybe somebody will be able to pick up an antigen that'll do the trick, but no. And I'm glad we had an opportunity to bring that up. This is, in terms of the immunity, this is a much more complex situation.
Yeah, and that's why we're using the entire virus, right?
Yeah,
we don't
absolutely.
really know. Yeah, I mean that's why the 2 polio vaccines we used the whole virus, because when they were developed, we didn't have any idea what.
Are there any subunit vaccines for viruses?
Wes,
yeah. Well, the mRNA vaccines.
mRNA vaccines.
Well, that's true, but that's.
No, it is true. You can't argue around
pretty
it.
Recent. That's pretty recent.
Yeah, there's Shingrix is a subunit vaccine.
Shingrix, the HPV vaccine is essentially a subunit vaccine, and then it's one protein, but it is assembled into a virus-like particle, but it's essentially a subunit vaccine.
And what about
polio?
HBV?
HBV, yeah,
that's
sure.
right.
That's the surface protein.
Exactly
Could
right.
polio become a single subunit vaccine?
So you could, well, you'd have to make a capsid like HPV, right? But you'd have to produce the capsid proteins, and the problem is that they have to be processed by a protease in order to fold properly. So it's a little complicated. But there's some development going on, because not for polio, but for other similar enteroviruses, people want to make subunit vaccines. Yes, so we may see it in the coming years.
This gets at Rich's variety point in a different way, that there's a variety of viruses, and there's a variety of ways that we need to try to address making vaccines.
Yeah.
And I wanted to add to that, there's a variety of virologists also. They're all different.
You guys are siloed.
I don't know about that. We go to meetings.
You
we
have
talk
your
to
own
each
meetings.
other.
You have a meeting for polio. You have a meeting for, you don't get together and have a general virology meeting.
Of course we do. What?
I take exception to that, Dixon.
You do, but
in a big way.
How many people of all the subunit virologists attend your meeting?
A lot.
That's a complicated question.
Yeah, but we had over
right?
Had over 1,600 people, 1,800 if you include the online people.
Yeah, but you have workshops that are virus specific that are dozens of different viruses. It is true that some subgroups of viruses who will remain unnamed have a tendency to be sufficiently siloed so that their attendance at the ASV meeting is less than what we would like. But
there are, besides ASV, there's the International Congress of Virology, which attracts many different virologists.
Yeah,
there are, for example, the positive strand RNA virus, there are negative strand RNA virus meetings. So there are all kinds of permutations, Dixon. And then, of course, there's the herpes virus meetings and the retrovirus meetings and the picornavirus meetings.
Is there not a meeting for every virus type? Come on, there is.
Yes, ASV and...
No, the bacular guys don't have their own meeting anymore, right?
Oh, no,
right.
Really?
Oh, for each one. No.
Right.
But Dixon, in parasites, there's not a Trichinella meeting?
Yeah, of course.
Well, okay, so you're siloed too.
But then I didn't say we weren't.
Okay, you didn't, but it sounded like you were pointing out something.
No, I'm not being critical, and I want to build on what Rich just said, because if you want variety, just go to a parasitology meeting and you'll see it in spades.
Well, apparently I am going to one in October. So
yes, you are. Well, that's not just a parasitology meeting. They also talk about viruses at that meeting as well.
Oh, it's ASTMH, right?
Tropical. Yeah, a lot of arbovirus work going on there. So you'll hear about that. I'm sure you'll hear about monkey virus too.
Monkeypox?
Monkeypox.
Speaking of polio, go ahead, Rich.
I was glad to hear Michael say that he was taken by surprise too. Mm-hmm. You know, because
I don't know how you would know, right? Who would know?
Well, you know, it's on the flip side was the swine flu in air quotes outbreak during the. What was it? Was that Jerry Ford?
Yeah, that was Jerry Ford.
I can't remember.
Yes.
Where there was an outbreak on a military base.
Fort Dix.
And we went bonkers and made a vaccine, started vaccinating everybody, and it never got off the base. Never got off the base, and there were Okay, so there are counterexamples where you're right on top of the problem, and
right.
it turns out to be unnecessary and caused more harm than good.
You're
so
right.
It's hard to do it right.
One can argue for the first 2 months of COVID, we didn't do anything.
Exactly.
I mean, many people thought, oh, it's going to be like SARS-1, it's going to peter out. And I think the first country where it was picked up, China, didn't really move on it for a while, right?
No, they did not.
I think in my mind, at least, what really tipped that one was not only the spread to other countries, but a clear demonstration that it was spreading asymptomatically. That just changed the game entirely.
Yes. Mm-hmm. But, going back to swine flu, I was in Peter Palesi's lab, a PhD student in influenza virus lab, during that outbreak. And he used to say, this is a pig virus. So we didn't have any genome sequencing in 1976. But what he had that few others had was being able to fingerprint the RNA, right? He cut it up with RNase T1, and then he labeled the small pieces of RNA. He could run it in 2 dimensions and see spots. And he would say, Vincent, this is a pig virus. It's not going anywhere in people. He was certain it wasn't going anywhere, although who knows? In the end, who knows, right?
Who knows?
So, in hindsight, he was right. But, and Edwin Kilbourne, who was chair of the micro department that I was in, he and Peter were like this over this, because Kilbourne was the reason we made the vaccine and started immunizing people. He told whoever was responsible, yes, you should make this vaccine. And to this day, well, he's dead now. He's excoriated, right, for that.
He's recycling.
When he died, I wrote a blog post about him because he had an effect on my career that I very much appreciated. And I didn't mention the swine flu. And his family actually asked me to give a eulogy at his funeral because they said everyone else is criticizing him after he's dead for the swine flu thing, and you're the only one who appreciated it. So it was very nice. I was just surprised they found the blog post, right?
I wouldn't leave Maurice Hillerman out of it either.
Anyway, speaking of polio, I wanted to update us because a few weeks ago we talked about here on TWiV a case of polio in Rockland County in an unvaccinated individual. And it was turned out to be vaccine-related type 2 Sabin polio. And then they started looking in wastewater, initially in Rockland and Hudson counties of New York. And they found by PCR, as I figured they would, type 2 Sabin. And now they have just announced today that they have found it in New York City sewage.
And London.
Of course, it was in London some time ago, and it's also in Israel some time ago, because they look, and I will predict that, and I've been speaking with reporters all day today about this, I would predict that wherever you look in the U.S., you're going to find it, at least in big cities. And I think the only thing you need to worry about is whether you're vaccinated. Get vaccinated if you're not. That's it.
So the stuff that they're picking up out of the sewage, is it a variety of different genotypes? Okay, because we talked last time about how these viruses have a fingerprint, if you like.
Right.
Okay.
Right.
With polymorphisms. Is this a mixture of polymorphisms, or is it related to the case that was discovered? Do you know?
No, it's clearly related to this viruses circulating in London and in Israel. Right.
Okay.
Yeah. So someone traveled from one place to the other, and that's what's depositing it. Because, as we said before, most of the world uses oral polio vaccine, and that's upon reproducing in the gut, the virus reverts and it's excreted and it persists. And even if people are vaccinated, the virus will infect your intestines and you will shed it and spread it. And that's how it got to the U.S., even though we don't use OPV. And I'm sure it's throughout the U.S., and this is nothing new to this particular year. This has been happening for years, I'm sure, but we never looked for it.
Right.
And we just started to. I don't know why, because the CDC has always been refractory to looking for it. But, you're looking for SARS-CoV-2 in sewage. You can easily look for
Well,
an example of one case equals an epidemic.
What did Mark Blanche say? If you find it in the sewage, what are you going to do, right?
That's what he said 10 years ago.
Yeah.
He said, we don't look for it because we don't know what to say about it. And I was talking to Amy Rosenfeld about this, and I said, I wonder what Mark thinks now. And she said, he'll just say he's retired. That's what he told me when I asked him. He didn't want to talk about it. Anyway, so
what I was just reading this article that you put in, and they point out that they won't say where in the 5 boroughs of New York this had turned up. And then when you were mentioning that, it would be interesting to know from those samples, for instance, that John Dennehy, Davida Smith, and Monica Trillo had collected, if they went back in those, how far back might they see it in any particular
I asked
site.
them. I sent them an email a couple of weeks ago, and they said they were ordering the primers.
Right.
So they should have a storage of many months of samples, right? That would be very interesting.
I think would be cool.
4 years even.
And also, Davida Smith, who's now in Texas, she said she's got permission to collect local sewage, so she's looking there. So and I don't know, I don't know who actually did this work that's reported in the Times, if it was Department of Health or CDC. No, the CDC is here in Rockland County. They're investigating. So we'll probably be hearing more.
Yeah, stay tuned.
But I was asked to be on CBS News tonight at 7 p.m. live, and I said no because I'm on the train at 7 p.m. I'm sorry. And if I stay here at the incubator to do it, I'm not getting home till 10 p.m. because the trains are few and far between after. And you know what? We got TWiV, and we're talking about it here. So just listen to TWiV, folks.
Yeah, and there's other people who would be perfectly happy to get on CBS News and say stuff about polio.
Yes.
Maybe CBS would have paid for a car for you to go home.
They might.
Well, it's funny. And back in February 2020, I went on CNN for the COVID business, and they sent a car, picked me up at Columbia.
Oh, yeah.
And then afterwards, he brought me back. It was very nice. Yeah.
Yes.
Yeah.
But they didn't offer.
Should have demanded it.
I don't know if they want people in studio yet, Kathy. That was going to be by Zoom, right? Which
right. Yeah.
But that's fine. They can find someone else, and there will be more opportunity. But I did speak to a lot of other people today about this issue.
Good.
And I'm sure you always mention TWiV.
I don't always, because if it's not live, they're just not going to mention
but we've got
it.
archives that address the problems, right?
But I don't know. The guy from CBS called me Professor Racaniello. We would like to have you. So he somehow knows about me, right?
Mm-hmm.
Well, you're getting up there, kiddo.
I don't think so, but somehow he knew I was a professor at Columbia. I mean, there is a publicity
They can
department,
look you up.
And they go to them, and they often give them my name, but it's fine. Actually, this week someone. This is very interesting. So at 5:15, I'm speaking with Jeff Kluger, who wrote. He's a Time magazine writer who wrote. What's the name of that polio book that he wrote?
Yeah, the name's familiar. Yeah.
The special recipe, or
a splendid.
Yeah, the splendid something.
Something. Yeah.
Kluger, K-L-U-G, or Splend.
Maybe it's not Splendid. Splendid Solution.
The Splendid Solution. Jonas Salk and the Conquest of Polio. Anyway, I'm talking to him.
As opposed to Ted Williams, who was called the Splendid Splinter.
And initially he had asked Ian Lipkin, and Ian said, you should talk to Professor Racaniello. He's the polio expert around here. Around here at 168th Street. So that should be fun.
Everywhere.
That should be fun. All right. Let me see what we have here. And I think these emails are long, don't you think, to start any of them?
Yeah.
There's not that many. I think you only have one round in here, right?
Really?
But it's up to you.
I just need to finish at 5:15, that's all. Well, let's start and see how far we get.
Yeah, we don't have to. If we don't get through a whole round, fine.
Okay. All right.
I won't be hurt.
Kathy, can you start with the first one?
Bob writes, Dear TWivers, just to break with the usual, I will start with the however and close with platitudes. You can assume the best. I'm writing from my office on an inpatient ward in a rural Ontario hospital. As I came in this morning, I double-checked that among our 25 patient rooms, there are only 2 negative pressure rooms. That is, the beds in which one would bunk down patients who have an infectious disease. Based on current news reports, I would bet our team would reflexively send cases of monkeypox and COVID most quickly to those negative pressure rooms, assuming they were not already occupied, which they always are. Is the chronic high baseline strain on staff apparent? Which brings me to the attached screenshot from our Fed's COVID website. The chart illustrates our national hospital COVID occupancy over time, where you can see that July 2022 is shaping up much differently than both July 2021 and July 2020. I should note that the unusually high July 2022 occupancy exists despite the fact that Canada's vaccination coverage is very high. Over 85% have received at least 1 dose. Important to note for you that our constant hospital strain is not generalizable to the U.S., where your COVID occupancy is thankfully not an issue right now. But if it were, I would remind you of the MMWR report from 2021, predicting 80,000 excess deaths if ICU capacity were to exceed 100%. And he gives a link. I think that finding is generalizable to us in Canada. Also of concern for a province such as ours with a huge variance in population density, 80% ICU occupancy in Big Toronto might not be a concern, but I can imagine when ICU capacity even gets above 70% or so in a rural hospital such as ours, summer ICU staff will consist of generic licensed providers of all stripes, each a stranger to the next. All to suggest that from a public health perspective, hospitalization is appropriately the favored metric for determining where the pandemic is right now, at least in our province. As evidence of that shared sentiment, I'm also providing a link to our local health until wastewater surveillance charts, where you can see each station's RNA counts are graphed against its respective neighboring hospital's occupancy. And he gives a link. Platitudes. What can I say except to further note that my ward is a psychiatric ward and I am a psychiatric researcher with more than a passing interest in all things character. How ironic when I think of the finest examples of character strength and virtues demonstrated over these past 2 years, I most times turn to you TWiV virologists. Keep up your fine modeling of what it means to be both great scientists as well as great world citizens. Humanity needs more of you. Stay safe, Bob. Really
what a
nice.
Wonderful way to end a letter.
It's very nice.
Yeah.
So I understand that hospitalization is a metric for determining where the pandemic is, but I don't think it's a metric for measuring vaccine efficacy. That's a different story, and that's what is often done. And I think Paul Offit had the same reaction as well. But I agree.
This chart that he gives with comparing wastewater signal to hospitalizations is interesting.
Yes, it's very interesting.
It's not an exact correlation. There is a correlation. There are places where there appears to be a correlation, but it's not matching exactly. Interesting.
Mm-hmm.
Thank you, Bob. It's very kind. Mm-hmm. Dixon, can you take the next one?
I will. Steve writes, Hi, TWiV. When Vincent asked about big bands today, Dixon initially said there were some. Then your quick web search indicated all the most recent bands are from the 1970s. The reason for this is pure economics. The number of performers on stage, their adequate remuneration, and travel costs combined with the need for a big band to play in a venue that is neither too small, too few seats to make a profit, or not too big. A stadium is great for a fully amplified rock band, but unacceptable for big band musicians' aesthetics. Means the art form is economically not viable in our times. A big band of today, even one made up of living, still performing greats of the older bands like Chris Botti. Considering a tour of, say, 6 months out of the year, we'll always lose money on ticket sales alone. Therefore, a ban like this would have to depend on grants or private donations. Indeed, when any of us hear a big ban on limited tour today, it has been subsidized by one or both of these means. Compounding this last statement, big bans don't attract much grant money as they practice what is considered to be old and niche art form. But the niche part of this is only true because there are no classic big bands touring around these days. It is a circular and self-defeating economic conundrum. No need to read all this verbiage on the air. Too late. As a humble musician, I only hope to give back a little illumination from my field in return for an amazing wealth of virological understanding I have received from all of you and your guests on TWiV over the years. Kind regards, Steve.
That's actually very interesting.
Yeah, I didn't know that.
Yeah.
It's the economy, stupid.
Mm-hmm.
Well, Duke Ellington used to remark all the time in public that the reason why they're not playing in more places is that all of his gentlemen have to be fed. And what he meant by that was all the players in his orchestra. And he had like 18 people. And that's a lot of people to be responsible for.
Yeah. So when TWiV goes on the road, we're going to have to get a grant to subsidize it, basically.
We will. We get our money up front and we put it in a offshore bank account.
Rich, you should take the next two, Rich.
Okay.
It's short.
Mary writes, Dear TWiVers, thank you for all you do. I am gasping here in the back after trying to follow the Offutt machine gun delivery. You all responded by exceeding the speed limit to which I am now accustomed as a longtime listener. You were all stars on the episode. However, I don't really understand why we shouldn't modify the COVID-19 vaccine. We modified the flu vaccine every year on account of changes in the virus, so why not COVID? Only an accountant, Mary. Well, Mary, it's happening, okay? And I'm sure it'll happen more down the road. We can debate it and talk about it all we want on TWiV, and then the world goes on and does what it wants to do.
Well,
which is following on your logic.
I just want to quote Jerry Weir, who Michael knows from CBER. He said, "It's not flu. It's different. It's a different virus, and it's a different vaccine, and so we can't apply to one what we do to the other." But you're right; it's happening.
Jerry Weir, by the way, is also, initially at least, a pox virologist, went through Bernie Moss's lab. Okay?
That's correct. That's correct. And he is the person who hired Amy Rosenfeld at FDA. Yeah.
You might say, Rich, that nowadays he's thinking out of the pox.
Ooh.
Alan's not here. What do you expect?
That's good.
So take the next one, Rich, also.
Kim writes, Hello, TWiV. Do you think monkeypox should be a concern for people participating in close contact sports like judo, wrestling, or Brazilian jiu-jitsu? If so, should participants consider getting vaccinated? Thank you, Kim. I would say, yes, it should be a concern. That's a perfect mechanism for transmission. I would say that, yeah, you could get vaccinated, but I would also say that the transmission, primarily, as we understand it, is from lesions to other people. So if you got lesions, you don't wrestle, okay? Or do other things that involve intimate contact.
But you know that's not going to work, Rich, right?
Exactly. It's people. It's behavior. Right?
I go along with Kathy's cynicism. It's a logistic cynicism, but it's true. It's just we know humans don't do that.
So I don't know if you're going to be able to convince your local public health people in the absence of in the face of limiting vaccine supplies. But yeah, I would say that people involved in those sorts of close contact sports, in particular wrestling.
I had to look up. I just had to look up Pollyanna, and I found that it's an excessively cheerful or optimistic person. So now I understand what Kathy meant.
Aww.
I was accused of being Pollyanna in a faculty meeting. I will never forget.
But that's not a bad thing, Kathy. It's good to be optimistic.
It was not meant to be complimentary.
Oh, I'm sorry. Well, I would have viewed it as complimentary because if you're optimistic,
realistically jovial.
I think being optimistic is a good thing, right?
Yeah.
Anyway, if I hear right from Michael, by the end of the year, we'll have enough vaccine, and if you wrestle or do these things, you should probably get it, right?
When you say enough, though, I meant to say how much is that? He said 2 million doses. Does that sound like enough?
No.
I would say that if you're concerned about this, okay, and you can't get vaccinated, make sure who it is that you're interacting with.
Hear, hear.
Okay? As Michael emphasized time after time, this is there's a huge behavioral component in this, okay?
Right. Yeah, but there's examples where people have no behaviors that are consistent with getting infected, so that's the problem. It may be rare, yes. I don't know. All right, let's do one more here. We have Mark. Hi, Vincent, Dixon, Kathy, Rich. I want to thank you all for the wonderful, informative, and balanced podcast. I know of no other up to your academic and clinical level. Well, thank you. I just
stop
finished.
Right there if you want.
Yeah, I think that we should stop. I just finished the July 10th episode, Boosters On, with Paul Offit. I think Paul is a wonderful doctor and infectious disease specialist, as well as an excellent researcher. I worked under him during my infectious diseases fellowship at CHOP years ago. I love the discussion and agree with his views regarding the low risk of fully vaccinated individuals coming down with severe COVID illness. One point that was not brought up during the pod is the issue of asymptomatic viral spread or spread during or just after a mild COVID infection in the fully vaccinated. One may feel well or just mildly ill after infection, but your chance of spreading COVID must surely be higher than if you never were infected at all. As a 65-year-old fully vaccinated and boosted ×2 pediatrician, I'm not concerned with becoming significantly ill myself, but I keep awake at night with the fear of becoming infected and inadvertently spreading disease to my patients and their families. For this reason, I have not lowered my guard in avoiding indoor gatherings if not necessary, and I always mask in public when a public gathering is absolutely necessary. Perhaps you could address the little data available regarding infectivity of those with COVID after full vaccination, even those only mildly ill, and the risk that those individuals might inadvertently spread disease to their contacts. I put 2 references into papers that have been discussed by Daniel, and I think he did a good job of doing that, Mark, and you should listen to his updates. And there's a New England Journal and a Nature Medicine paper, which are not perfect, by the way. They're flawed because they don't measure infectious virus.
And they're from TWiV 916.
All right. I understand we are all tired of this pandemic and numbers have fallen significantly, but when the U.S. daily death rate hovering 500 a day is the equivalent of a jumbo jet crashing to the ground with all hands lost daily and is 5 times the USA daily death rate of automobile deaths, gunshot deaths, and influenza deaths, about 100 a day, I can't see how we get back to normal and accept those as part of routine life. I'm still a strong believer of primum non nocere and don't want to contribute to the problem if I can help it. I just don't know when, if ever, we will be able to get back to a normal life. So I, again, refer you to Daniel, Mark, who says that these deaths are in the vast majority of those 500 deaths a day are in unvaccinated people, immunosuppressed people, people over 75 who have been vaccinated. And the solution is to early on after testing, give them Paxlovid or treat them with Evusheld prophylactically. And he said the problem is not all doctors do that. They say, let's wait and see what happens, and then it's too late. So in his opinion, all of these deaths are preventable with monoclonal and antiviral therapy, or at least most of them. And so we can get back to life if the doctors in the U.S. would just get with the program. And I can't tell you how many emails that he gets of people saying, I tested positive, I'm 85 years old, and my doctor said, oh, you have a mild infection. Let's see what happens. We won't treat you yet. And then when it gets worse, it's too late. So I think it's a complicated issue, Mark. I understand. But I do think, according to Daniel, those deaths are preventable. All right. Let's do some picks of the week. Dixon, what do you have for us?
I got 2. I'm going to add to my jazz big band picks. This is big band number 4. Duke Ellington and his orchestra. He's was a remarkable musician, band leader, worked out of New York City. In fact, died at Columbia Presbyterian Hospital. I actually knew the resident who performed the autopsy. He was a trumpet player, by the way. And he was like in shock the next day he came in after that. He said, "Guess who I performed an autopsy on?" And I said, "No idea." I said, "Duke Ellington." Wow. The signature song that everyone thinks of when they think of Duke Ellington is "Take the A Train." Vincent, how many times have you taken the A Train?
Hundreds, thousands of times.
Thousands of times to get to the downtown campus of Columbia. No, that's the number 1.
No, that's the
number
one.
2.
Yeah,
that's right. You don't take the A Train.
No.
You go right past that. You go on to 59th
but if I
Street.
want to go to Columbia from here, I would take the A Train.
That's right.
From
It's
Penn
fast.
Station. It's the express. It's fast. Yeah.
That's right. And if you listen to the words, you'll find out why you should take the A Train. The second pick happens to fall on the anniversary of a 1958 event that occurred in Harlem in which a photographer wanted to take a picture of as many local jazz greats as was willing to show up. And he put out the word to a few of them and said, "Please meet me." And he gave them an address. And they started to show up. Slowly at first. Then they went back and they called their friends. "You got to get over here. You can't believe who's here." And the next thing you know, they had 57 of the world's most recognizable great jazz players on one stoop in Harlem, getting their picture taken. Now today on WBGO, which is my jazz station go-to out of Newark, they were playing the signature song of each one of those 57 people.
Oh, that's
and
cool.
It was absolutely riveting.
That's
I almost didn't make TWiV because of it.
It's a great photograph. I love it.
It's a great photograph, and it's just remarkable.
Yeah.
What a great premise for a show.
And what a great
Yeah.
premise for a show.
Yeah.
That's right. That's exactly right.
And
Nice.
it's really good, too, that they identified all of them.
Yeah.
In
They
this photo.
did,
Yeah.
and there
Yes.
was a documentary made on this.
Nice.
And you can, I guess, get it on YouTube. I'm not sure. And each one of those people established modern jazz in the United States.
Nice.
That's what they did. Yeah.
By the way,
That's why I'm so passionate.
TWiV 226 Dixon was taking the viral A train with Terry Dermody. It was you, me, and Terry.
Dermody, I remember him. I remember Terry Dermody.
Well, he was a medical student at Columbia, and
Yeah, he was.
I think
Yeah, absolutely.
he took your lectures. He knew you.
Yeah.
So it was you and me and
remember
Terry
him.
in my office, and there's a nice photo there of you and Terry.
Oh, how cool. How cool.
I'm not sure why it was taking the A train, but maybe he took the A train to get from his hotel to Columbia, right?
That's right. You take the A train if you want to get to Harlem. That's the words of the song, and then it goes on from there.
You don't think it was something about viral trafficking?
Yeah, it could have
That's the way
been.
I interpreted
Might have
it.
been. It might have been mentioned in it. Yeah, he may have said, "Yeah, the way this virus moves is like the A train." It could have been it.
All right. I have one more big band to go through on our next TWiV, and then we're off on the smaller groups.
All right, Kathy, what do you have for us?
I picked YouTube that my brother sent me entitled "How the U.S. Postal Service Reads Terrible Handwriting."
Yeah, that's great.
And it's really fascinating. Basically, OCR gives us 99% of envelopes being read, and then the 1% that are smeared because they got wet or they're just really bad handwriting, they go to this now only one facility. There used to be dozens of them, but there's one facility in Salt Lake City, and it goes through how they read them, what they code in, and how much time they have. And before the letter is even dumped off, if they get the result back fast enough, then it sends the letter on its way. And only about 0.5% of them don't totally get handled that way and have to really be looked at by a human being and then resorted.
Cool.
But it's just fascinating.
That's
Amazing.
great. I love it. Write clearly, folks.
Yeah.
Rich, what do you have for us?
So this has been making the rounds, so many people might be familiar with it. But this is CBS News report of an incident where a prominent French scientist named Etienne Klein posted what on Twitter, I think, what they claimed was an astonishing new image taken by the James Webb Telescope of Proxima Centauri, the star closest to the sun, and there was a picture there. And then confessed sometimes later that, no, actually, that was a slice of chorizo. Okay. So, yeah, unfortunately, is Etienne... I'm sorry. What sex is that?
It's a guy.
It's a guy? Unfortunately,
I should say
he
most
got a lot of
commonly.
grief for okay. He got a lot of grief for this. Okay. I thought it was a hilarious joke. He got a lot of grief from it because people got upset and stuff. But he says, okay, I apologize. But the point is, look how gullible you are. Okay.
No question.
That's great. Wonderful.
I took a histology course once, and for the final exam, the instructor had a microscope set up for extra credit. And you walked past and you looked in, and you've never seen anything like it. I said, what the hell is this? It has all kind of bizarre shape. It was a slice of bologna.
Yeah. All right. My pick is the website for the Bat Conservation Trust. I found this at the Bat meeting I went to. So this is an organization, a nonprofit in the UK, that is providing information and assistance about bats. They have lots of information, types of bats, where they live, fun facts, what they're doing to conserve bats. There's a helpline if you find a bat in your house, what to do. You can donate. They have events of various sorts, meetings and so forth. And if anything, just the picture on the homepage is just gorgeous. This lovely bat, right? Looks like 2 sets of ears. It's great. But a lady from this trust was at the Bat meeting, and she was wonderful. And you should check out a lot of good information here. Bat Conservation Trust.
Cool.
And we have 2 listener picks. We have one from Bill, who says this is particularly for Vincent. This is an article in The Atlantic called "The End of Manual Transmission. Stick shifts are dying when they go. Something bigger than driving will be lost." And this is by Ian Bogost, and I can't agree with more. This is a person who drives a stick shift and bemoaning their going extinct. Bill says, over the many years I have been a TWiV listener, I can't count the times this topic has come up. Never gets old. You're absolutely right. And I still drive a stick shift because you can still buy them. At least I was able to find. I bought a used Mini Countryman or whatever it's called.
Mini Cooper?
Yeah, something like that. Yeah, Mini Cooper Countryman. Yeah. And it's got a 6-speed in it, and I'm very happy.
Are they made by BMW now?
Yeah, they are.
Things?
They
Okay.
are. And they have the same classic 6-inline engine, which is it's perpendicular to the drive shaft. Very cool.
Well, we have one stick shift left in the extended family, which is that Beamer that you're familiar with, Vincent. I think it's basically the same as yours. It's 2007. My daughter's now driving it. So
it's a 2007. Mine was a 2012. But I remember your wife gave it to someone else, right?
She gave it to Sarah.
Sarah.
So
Very cool.
we still see it. We still see it almost every day.
So she likes to drive it, right?
Oh yeah, she drives it to work.
Good.
Yeah, I probably said this before, but when my kids learned to drive, I insisted they learn to drive a stick. They learned on an old Toyota Camry, and they complained and complained. Okay, and now they, in retrospect, thank me. Okay, because it was so cool.
Well, my son, I taught him how to drive on a stick, and he has a stick shift now, but the others don't. But the other 2 kids. But I put 250,000 miles on that Beamer, driving to Columbia every day, and I I traded it in for the Mini Cooper, and
you did. Oh,
and it was really.
that.
I mean, they offered me $600 for the trade, and I said, "I'm sorry, I cannot take less than $1,000. I'm going to walk away because just emotionally, I can't handle giving my car to you for $600." And he said, "Okay, fine, $1,000." But it's sitting there in the lot. They took the license plates. I just was so bloody sad to leave that car there because they're going to just break it up for parts, right? It was. I spent so many hours of my life in it.
BMW recycles all their parts. That's what they're famous for.
I was just very, very sad.
You should have asked him if you could keep the shift knob.
And then last year, my son Aiden, who drives a stick, he says, we should have kept that, and I could have played with it and tinkered with it. I said, yeah, you should have. Why didn't you tell me before? I would have kept it, Wouldn't have to be destroyed, but it's gone, and I have... I don't like to think about it. I know it's stupid because it's a bloody car, but I spent a lot of years of my life, and listening to podcasts in that car got me into podcasting. So
we got it.
Anyway. And then we have another listener pick from Jay. I thought you all might like this. This is a great video from Nick Lane on how the Krebs cycle powers life and death. Really good. Nick Lane is a biochemist in the UK. He's written a bunch of books that are really good, like Oxygen and so forth. And I highly recommend them. And this is really good. So
I probably ought to watch this because the Krebs cycle's always been a mystery to me.
I watched it just before our TWiV show. It is remarkably well done and remarkably well taught and remarkably understandable.
Yeah, Nick is great. He's really good. Got to get him on one of our pods one day. All right, that's TWiV 927, microbe.tv/TWiV for the show notes. Your questions, comments, and picks go to twiv@microbe.tv. If you enjoy our work, please support us. We depend on your support. Microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Dixon Despommier is at trichinella.org, thelivingriver.org. Thanks, Dixon.
You're very welcome.
And Rich Condit,
good stuff.
Rich Condit's an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. This just never gets old.
Well, you're quite right.
I must be really a nerd. It
doesn't get old, but we do.
It's good stuff. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.